Good morning, students. Welcome back to Open Web Based for Learning platform. Myself, Dr. Padma Sundari, Assistant Professor, Department of Botany. We will start with another topic of vascular cryptogams known as heterospory and seed habit. The first terrestrial plants appeared in the Silurian era, and so pteridophytes and ferns were born. They are devoid of seeds. Reproduction via several spore species. Speeds are not generated after sexual reproduction, but zygotes are. Along with the emergence of heterospory features connected with seed behavior, the pteridophytic plant Selaginella was evolved. It is believed that heterospory is necessary for the production of the seeds. If you see the definition of heterospory, hetero means different. Two different kinds of the spores produced by the same kind of the plant differ in terms of number, size, structure, and developmental pattern. And this type of spores, heterosporous, and the plants had heterosporous plants which are found in vascular cryptogams. On the basis of number, size, and structure of spores, they are of two types. One is known as the homosporous plant, and the other is known as the heterosporous plant. In homosporous plants, only one type of sporangia and spores are produced produced in the same plant. Example is the Lycopodium species. And here, the characteristic feature of the homospores plants is the development of the sporangia is eusporangiate type, and the spores that are developed in the sporangia are similar in size. Eusporangiate is a condition where group of cells will develop the sporangia. In heterospory, spores are of two types, which are different in size. Small size spores are called microspores, and the sporangia they are produced are known as microsporangium. Larger in size spores are called megaspores, and the sporangia they are produced are known as megasporangium. Male gametophyte of the germination of microspores, and the female gametophyte is formed after germination of the megaspores. According to Rashid, 1976, only 9 genera of the pteridophytes are heterosporous, where you can see Regniladium, Marsilea, Salvinia, Azola, Selaginella, then Platozymum, Isoetes, and Phil- Pilularia, and then is the stylites. The characters of heterospory, mostly they are leptosporangiate type of spore development. In leptosporangiate, the spore develops from a single initial cell. The entire sporangium develop. There are two different kinds of spores: the microspores, which are more numerous, and the megaspores, which are less. They get larger in size because they are receiving more nourishment. So you can see the megaspore and the microspores. Re- Regarding the origin of heterospory, it is acknowledged that heterospory is derived from the homospores condition, which is primitive, based on the evidence presented in paleobotanical evidences, based on the data from the research on development, and indication from the research experiments. So, if you see the significance of heterospory, there is distinctiveness between gametophytes. The formation of a seed habit is very crucial. Produce gametophytes that are endosporic. The developing embryo point of origin is provided by endosporic development. Plant may now flourish in a variety of environmental situations, not just aquatic ones. Th- The phenomenon known as seed habit resulted from the adoption of heterospory and the retention and germination of single megaspore within the megasporium to generate a female gametophyte. One essential and comprehensive requirement for production of the seed is heterospory. Regarding the seed habit origin, only one megaspore develop in each megaspore angium in heterospory decreases in the male gametophyte size. There is a creation of the integument, an extra layer of the covering. Throughout its life, the megaspore is present inside the megaspore angium. The nourishment is supported to the fetal growth, and the apical portion of the nucellus is established to accept pollen grains on microspores. Selaginella is used as example to show how pteridophytes evolved to become seed-bearing organisms. The occurrence of heterospory phenomena, megaspore germination, occurs inside the megasporangium. A single megaspore developing inside the megasporangium, megaspore retention inside the megasporangium for the whole of its life, such as Selaginella monospora and Selaginella rupestris. While all these symptoms stop the formation of the seed habit in Selaginella species, the seed cannot fully mature into a seed in the traditional senses. Regarding the origin and development of ovule, the ability of the spermatophyte to produce seeds is their most important characteristic. The ovule must mature before seeds can begin to form and develop. Numerous theories were put out to explain the formation of evolution of the ovule. The concept of telomere, according to Smith 1959, Long 60, and Andrews 1960. The Rainier type branching telome is a representation of the initial stage of development. A few of them are viable and produced. First, there has been a decrease in megaspore counts. The megasporangium upper portion increases in length. There it was decided to build the structure that collects pollen grains. Sterile telomeres alternation led to the development of the immune system. A structure resembling a capsule developed as a result of partial fusion of sterile telomeres. Capsules were originally separated from the nucellus, but subsequently the basal part of the capsule become attached. They are found in the plants of gymnosperm lanterns. Second integument evolved by linking of the sterile telomes together, as it in case of gymnosperms scotica, and in Calathospermum scoticum, the nucellus and the integument are connected. Because of the lobe extinction and a decrease in the quantity of the anterior lobes, when integuments unite, a micropyle is formed. Rather than the front of the body, the micropyle was there. The process of collecting pollen grains began. Number of integuments and cupules lobes shrank even further, and in the end, both the integuments combined. As a result of all procedures and the steps that follow, and the majority of the fossil records are now available support the telome idea. It was become extremely well developed. The significance of seed habit is the shield of the embryo from unfavorable environmental circumstances. It provides nourishment, it pays parental attention to the growing embryo, and the possibility of hibernation takes place, and there is efficient distribution of the seeds. Thus, we can draw the conclusion that the development of seed habit and heterosporous conditions are related. The formation of the seed habit is significantly influenced by Selaginous heterospory, and higher plants may have evolutioned their seed habits as a result. Thank you.